Hello and welcome once again to Concepts of DNA. One very interesting evolutionary mechanism which the cells have maintained in themselves is repair of damaged DNA by photoreactivation. I'd like to tell you something about the Earth's evolutionary history before actually venturing into the molecular level of how this thing happens. 2.5 to 4 billion years ago, when this Earth was a hot ball of fire and gradually it cooled down, in those times in the sea there were certain bacteria, the species who were continuously producing oxygen and free radicals. Try to understand, most of the oxygen which came out of the sea, and it, after some billions of years ago, it created the oxygen atmosphere. Those times, it is called as the oxygen boom or the oxygen evolution era. Means there was such a huge amount of oxygen produced. Before there was an anaerobic condition, all the anaerobic bacteria, they were constantly working, and some archaebacteria, maybe the bacteria in the sea, and huge amounts of oxygen was being produced, and the oxygen burst on this earth created a situation where free radicals were there. Means from a reductive phase, reduction reactions were going on, and suddenly oxidation reactions started. So this was toxic for most of the species, cellular species, which were just trying to evolve on that time. So what happened was, if you see, in those times it was very necessary for the cells, if they wanted to survive, to show some sort of repair. Primitively, bacteria are the only species which survived in those times. They continued to survive, multiply, live, and change the whole scenario of the biophysico-chemical status of the earth. And today we are what we are due to the activities of the microorganisms 2 to 4 billion years ago. So the point is, from that time, and when the oxygen layer was formed, ozone was formed. Ozone was not there, high amount of ultraviolet radiation used to reach the earth. So if a cell had to survive, it had to show some mechanisms for repair of its DNA and survive those harsh conditions. Means on the ground there were free radicals, and there were oxidative species, that is reactive oxygen species, which you have seen in the mutagenic agents, and on the top there was ultraviolet light coming heavily from the sun. Interestingly, sun was 30% less hot than the time which is believed in this era, but still ultraviolet light was more, and on the earth reactive oxygen species or the free radicals were more. So the bacteria in the course of evolution developed a mechanism by which it could sustain these harsh conditions. So if you just see here, the DNA damage, it could be repaired, or it will be defective, it will not be repaired. If it is repaired, the cell will have genetic stability, as the microbial cells, they have been maintaining throughout the course of evolution. And if there is a defect in the repair, or it is incomplete or is not repaired, then genetic instability will either lead to diseases in the multicellular system, or genetic divergence will be there from its original hereditary characters, and it may be a evolution or it may be a cell death. So the damaging agents, we have seen this earlier, are, if you just see, there are four types of various types of agents, and there are that many types of repair. So the first we saw X-rays, oxygen radicals, alkylating agents, etc., which can give rise to conversion of bases to uracil, abasic sites, that is depurination, 8-oxyguanine we have seen in the mutagen types, then single stranded breaks in the double stranded DNA, double stranded break in the double stranded DNA, then damaging agents like UV light, polycyclic aromatic hydrocarbons. This can give rise to bulky addicts, and purine that is dimers, then X-rays or the anti-tumor agents also for that cause. They cause interstrand cross-link, double-stranded breaks, and replication errors, which are very common. That gives rise to a AG mismatch or a TC mismatch, and a insertion or a deletion type of mutation. All this can be repaired. The first type, it can be repaired by base excision repair. Second, nucleotide excision repair. Third, recombination repair, which has a homologous recombination event facilitating the repair, or a end joining event, that is the non-homologous end joining, NHEG it is called as. That also this is for your higher studies, not presently mentioned in your syllabus, but those interested, as I say, there is no limitations to the syllabus. It should be, you can venture the end joining recombinational type of repair. Actually, recombination repair is not there in your syllabus. It's very interesting, where the recombination event, it facilitates the removal of wrongly incorporated base. Mismatch repair is also there, and this repair processes are a part and parcel of the cells system, where DNA is very prone to damage, whatever the function is there. But see the beauty of the DNA molecule, that it, first of all, it replicates itself in a very foolproof process, and secondly, it does undergo a vast, huge errors in the process of whatever it does, replication, transcription, translation, but it has a tendency to repair. So all organisms, they possess repair mechanisms to correct the errors. DNA polymerases themselves, they have a error correcting property. In addition, there are some specific repair systems in prokaryotes and eukaryotes. The repair system which is there in these cells, it is made up of a system where it detects the defect. First of all, enzymatically it will detection mechanism is there. It removes the defective portion of the DNA. This is what you can say the modus operandi of the repair system in all the cells. That is the repair system. It comprises of elements which will do this in stepwise fashion. First, they will detect the defect. Second, they will remove the defective portion of the DNA. They will add the correct nucleotides in the gap. They will ligate, that is ligation of the newly added nucleotides with the pre-existing strands. This is stepwise process carried out by certain proteins and enzymes. There is a huge array of proteins for all the repair systems, which include, there are 4 which we are going to study. Today I will be just explaining one, because that is totally separate from the remaining 3 to 4 which are there. So today I will be dealing with photoreactivation. The remaining types are mismatch repair system, the base excision repair system, nucleotide excision repair system, and the double strand break repair system, or you can call it as the homologous strand exchange repair system. All this I will be dealing in my next lecture, but today we will be dealing with the photoreactivation. But before going to photoreactivation as such, we will just see what actually is the concept of repair. What you should understand by studying these 3 to 4 types of repair. You should know what is a DNA repair system. You should know what DNA damaging agents they do. You should know the concept of DNA lesions. Anything which is damaging a DNA and a addict or a dimer or some base change, or which is hampering or stopping the process, is called as a lesion. Different DNA repair mechanisms in the cell. You should know the different types of repair mechanism in all the types of cell, and you should know that this repair system which was discovered had got a Nobel Prize recently, that is 2015. Till now even the works are going on with the repair system of the DNA, and exclusive and very interesting things are being opened up. So this is what you should know. So what is a DNA repair? DNA repair is a damage to the DNA which occurs is reversed back. So DNA is a genetic information you can say the storehouse of genetic information of a cell. Okay, it is the headquarter of all the phenotypic and genotypic characters in the cell. Most important biomolecule which is essential to retain the genetic information which should not change. So therefore the DNA replication is foolproof in nature. Chance of DNA damage is very high because of the processes, as you know, 5,000 to 10,000 bases in a human body, they are constantly replaced every day, and even in a eukaryotic system, you know that 1000 in 1000 bases, one wrongly incorporated base, it exists. So DNA replication errors are there, and DNA recombination errors are there. So this repair mechanism overcomes all this burden on the DNA. So DNA damage in humans or in somatic cells causes cancer, loss of gene function is there. Okay, what does this repair, if it sustains, what does it do? It damages the gametes, it is transferred to the progeny, so whatever the defect is there, that is passed on to the next generation. Damage to the DNA is a major threat to the metabolism, so it is a major threat to the life of all organisms. So most of the cells, they have a mechanism to rectify this damaged DNA to overcome the damage. So these methods are all the methods which are there, they are collectively known as DNA repair mechanisms. So DNA repair is a very essential process for the ultimate survival of the cell, the tissue, the organ and the organism. So as a whole, wherever there is an involvement of a control or involvement of DNA, it is there because it is the main thing which controls the metabolism. There is repair. Okay, so the destructive forces which are faced by the DNA in the cell, we have seen they are of various types. Mutagenic agents are of various types, but broadly speaking, if you see the attack on the DNA which is there and its force which is responsible for destroying, damaging, I will say, the DNA is of two types. One is internal and other is external. We'll call it as intrinsic factors or intrinsic activities being disturbed, and external forces which are faced by DNA in the cells. So intrinsic, metabolic intermediates, I have told you that there are inborn errors in the metabolism which results into base analogous compounds, or there are compounds which can cause other bases to change, like the dyes are there, they incorporate. So these are intrinsic reactive radicals, free oxygen radicals which are there, these are damaging. Then DNA replication and recombinational errors, these are taking place within the cell. So these are the intrinsic destructive forces which has to be faced by the DNA, and DNA has to overcome the repair. External factors, we are going to study the external factor today. That is, one is radiation, ultraviolet light. Ultraviolet light, X-rays, gamma rays, all almost have same similar effects, that they cause lesions in the DNA. Carcinogenic chemicals, that is mutagenic chemicals, and DNA intercalating agents, these are the factors. Why DNA intercalating agents is extrinsic? Because it is uptaken from the growth medium. The cells are not synthesizing these dyes. Okay, though the effect is intrinsic, the forces which as DNA is encountering is coming from outside. That is, dye, if it goes inside the cell, then it becomes a mutagenic agent. So all these chemicals which have an impact on the DNA, they create a damage in the DNA somewhat. Whatever the damage it may be a dimer formation, it may be a single stranded break, it may be a double stranded break. All these collectively, they are called as lesions. So for all these, there are repair mechanisms available. Repair mechanisms, DNA repair mechanisms can be categorized into 7 different areas. We are going to see 3 to 4 of this, and remaining you can study it in your PG or the postgraduate higher level studies. So today I will be emphasizing mainly only on photoreactivation, and my next lecture will be nucleotide excision repair, base excision repair and mismatch repair. Apart from that, double strand break repair are there, homology directed repair is there, and SOS response and DNA repair is there. SOS response is the most common type of response. Maybe in the end I will throw some light on that. So the first 4, if you see, which we are going to see, photoreactivation, BER, NER and MMR, for the sake of convenience they are cut into short and called likewise. So for these 4, they have only damaged strand of DNA is repaired and the undamaged strand acts as template. Means in this 4, the damaged part is only repaired, the undamaged part, it acts as a template for adding the correct nucleotide. Whereas in double stranded break repair, homology directed repair and SOS response repair, both the strands are repaired. So that is a separate category of repair which involves larger number of proteins. In the end of the slide, I will give you a total list of enzymes which are involved in all these types of repair. So we will see photoreactivation as a mechanism of DNA repair. So what is photoreactivation? Photoreactivation is a DNA repair mechanism where there is a repair of the damaged DNA, the damage which has been caused by ultraviolet light. So it can be defined as a DNA repair mechanism in which recovery of UV or ultraviolet irradiated damage of the DNA is reversed by visible light. This method was discovered by Aziz Sankar. Okay, means he got the Nobel Prize in 2005. But we all know the history, how various scientists they were. There are many scientists who were involved before Aziz Sankar got into the award. But this is a method which recovers the DNA after exposure to visible light. A method, first they are exposed to ultraviolet light, therefore there is a damage, and then they are exposed to visible light, due to which there is a repair. So UV induces ultraviolet light, it induces pyrimidine dimers to form, and visible light induces an enzyme called as photolyase, which removes the dimers. So this is the first DNA mechanism which was discovered, and major findings, the final findings, tens and hundreds of scientists were involved in the discovery. You go on reading the papers, you will find many scientists. The pioneering scientists are totally different, award winning scientists are totally different, but award winning scientists are those who make a breakthrough in the understanding, and this breakthrough in the understanding of visible light for repair was done by Aziz Sankar in 2015. He got a Nobel Prize. But we have seen that the major work was by Albert Kleiner in 1940s. So this discovery, as I said, it involved many scientists. In 1920s it was Hermann Muller, he showed the mutagenic effect of X-rays. Albert Kleiner in 1940s, he showed what is photoreactivation by use of visible light, and photoreactivation is an enzymatic process. This was proved by Stan Rupert. The whole last lecture was dedicated with his photo on the left of your screen, who was Stan Rupert, who proved the theory that photoreactivation may be an enzymatic process, a very important step, and he called it as photolyase. So what actually this ultraviolet light does? Now I am giving a total separate lecture on photoreactivation because I have two main motives behind that. My objective is you should be able to see photoreactivation as a separate and very important phenomenon in the bacteria, in prokaryotes. First of all, the rest of which are there, that is the nucleotide excision repair, base excision repair, mismatch repair, are present in bacteria as well as multicellular system, but this light induced repair is exclusively for bacteria, and E. coli is today studied thoroughly for this. So the thymine dimers which are formed by ultraviolet light, which gives rise to a cyclobutane ring here. As you can see, the red ring, it is between two sugars. Okay, this is caused by ultraviolet radiation, and the covalent bonds formed between the adjacent pyrimidine residues. As you can see, thymine mostly, adjacent pyrimidine residue, thymine, the residues can be not only thymine. It is very popular that thymine they form very easily, but cytosine and cytosine can also form covalent. They can, but they have to be adjacent to each other. So TT, CC and TC, these are the three probable dimers which can be a result of ultraviolet light induction, remember that. But out of that, as I said, thymine dimer is the most common form of dimer. All these attached to each other by the sugar, the two sugar molecules, they form a cyclobutane ring. Remember this ring is called as a cyclobutane ring, and such dimers, they are called as cyclobutane photodimers. You have to remember these terminologies. They are called cyclobutane photodimers, CPD, and the thymine dimer formation causes melanoma in humans. Okay, in the bacterial cell, it causes the stoppage of DNA polymerase. DNA polymerase will not move further on this dimer, it halts, and there is stoppage of DNA replication, ultimately may lead to cell death. So thymine dimer formation by ultraviolet light, as I can you can see here, cyclobutane pyrimidine dimer CPD is formed between the C5C6 and the C5C6 of two adjacent sugars. Okay, and this results into a kink, or you can call it as a dimer or simply lesion. This is a terminology, DNA lesion. Any damage in the DNA can be called as a DNA lesion. So what actually, how it works? What is the mechanism of photoreactivation? It is a light dependent process. Remember, ultraviolet light is damaging and visible light is repairing. It is a light dependent process. Photolyase is a flavoprotein. This is photolyase. It is a molecular structure of photolyase enzyme that is induced by the photon energy. Two chromophores, just like what is happening in the photosynthesis, just like photon energy is responsible for induction in the leaf. Okay, there are stroma in the leaf. Just like that, the two chromatophores in the photolyase, there are two chromatophores in the photolyase, and a fully reduced FAD. What does this do? Photolyase, two chromatophores in the photolyase, that is fully reduced FADH and folate group. These are the two chromophores, due to which the photolyase, it absorbs the light at 300 to 500 nanometers. Remember this thing. DNA is absorbing as 260, lower wavelength, higher energy, therefore getting damaged. At visible light, that is higher wavelength, low energy, it is able to absorb light due to FAD and folate, and 300 to 500 nanometer it absorbs, and it acts by local free radical mechanism, by the mechanism of electron transfer. This is the principal mechanism of photolyase functioning. Light dependent photolyase, which is a flavoprotein containing two chromophore groups, that is fully reduced. These are the two chromophore groups in the photolyase, FADH reduced negative and folate group. This is one active in the active site, and they are responsible for absorbing the light energy. So what happens when this is there? You can just see the steps in which the mechanism of repair by photolyase. Photolyase, it binds to the thymine dimer. As visible light is there, the electron, as you get, this is a photolyase enzyme, FAD, and the folate is there. So photolyase, it binds first to the thymine dimer. FAD, it absorbs the light. This is a photolyase enzyme. Visible light is absorbed by the chromophore group FAD. FAD gets excited. It releases an electron. This electron interacts with the dimer, and a charged radical pair is produced. That is FADH plus the electron. Okay, photon, you can say photon is there, and the ring is split by cycloreversion. The cyclobutyl ring is split. How it is formed? It goes reverse. It disintegrates. This D rings. It forms a ring. It D rings. Reversion is there. Cycloreversion, and connecting bonds, they break. Electron, it returns to the FAD. Photolyase leaves the DNA, and photolyse, it leaves the DNA. Photolyse comes on the DNA. Enzyme and substrate specificity is there. E plus S. What is the S needed for this enzyme? That is a damaged thymine dimer DNA. So it comes and binds at the dimer. Then once it binds, it absorbs light at 300 to 500. FAD present inside this enzyme, it gets excited due to absorption of photons. It releases an electron. It's excited state. That electron goes to the cyclobutyl ring. Cyclic reversion is there. As it formed means it is a planar, it forms a planar. In a ring structure, it forms a planar structure. It is removed, and the thymine dimer is removed, and they become normal. There is no excision of the DNA. There is no cutting of the DNA. Nothing. No ligation. Nothing. Only the thymine cyclobutyl ring is removed due to the excited electron from the FAD, and this ring is split. Process is called as cycloreversion. You can see cycloreversion. So these are the steps. You can just, and then in the end, the photolyse, it leaves the DNA. So this is how the mechanism of photoreactivation takes place. This is a very significant reaction going on in the bacterial cells. Bacterial cells, they have been a part and parcel of the evolution where on the face of this earth, remember I had told you there was huge amount of ultraviolet light before ozone was synthesized. Before the ozone layer actually was formed, huge amount of ultraviolet light came onto the earth. There were free radicals also. So only those cells which were capable of sustaining these harsh conditions by some mechanism. And what was that mechanism? It was repair mechanism. So what is the significance of photoreactivation today for all the cells? Photoreactivation, you can remember it as the first form of DNA repair, not only identified, maybe it was the first mechanism present on the face of the earth during evolution. I can believe that. Okay. This is the only method where direct reversal of DNA repair, damage takes place. Direct reversal means the cyclobutyl ring is reversed. Reverse cyclic structure into a planar structure is done. So direct reversal method of DNA repair. Then a template strand is not required, means if it was cut off and thrown away, there was a gap, then template and addition of nucleotides would have been taken place, but this is not there. No template strand is required, no removal of nucleotides is there. Photoreactivation is not a universal phenomenon, therefore I am giving a separate lecture for this. Try to understand, photoreactivation is one of the most important phenomenon in bacteria. It is not a universal phenomenon. It is present in archaebacteria, plants and some animals, many animals, but not all. It is not universal. Many animals may show it, but predominantly this mechanism is exclusive for prokaryotes. Completely absent in placental animals including humans. Means wherever there is a embryo, egg, mammals, you can say it's not present in mammals. Placental animals, they don't show this, because remember, once the ozone layer formed, once there was no ultraviolet light coming onto this earth. Try to understand this point which is there, completely absent in placental animals including humans. Why it is present in bacteria and some animals? Those animals which were surviving before ozone layer formation show repair, and we humans got evolved on the face of this earth after the formation of ozone layer. Means we were born when there was very less or no ultraviolet light on this earth. So God forsake, if there is removal of this ozone layer by some nonsensical activities by the humans, then humans will be the first to die, because we don't have repair mechanisms. These are some of the significant points which you should understand while studying the photoreactivation. So it is completely absent in placental animals including human, and humans, they use nucleotide excision repair to remove the thymine dimers. That is a method involving cutting of the DNA, removal, and the replicative and the recombinational processes are used in that. In our next lecture we will be seeing these types of repair, that is nucleotide and base excision repair. So there are various types, and all these types today you see here, see the focus on the laser pointer. So we saw direct repair today. Direct repair, it involves, I told you the mechanism of DNA photolysis here, but you should remember there are two more proteins which are involved in removal of the direct damage, that is damage due to O6 methyl guanine. We saw there is a methyl guanidine molecule which can form in wrongly incorporated bases in the DNA, that can also be directly repaired by O6 methyl guanine DNA methyl transferases. What does this enzyme do? As the name itself indicates, it goes there and just removes the methyl group, and methyl is transferred, so it reverts back to its normal Watson and Crick pairing, and it is what you can say, without cutting the DNA. And there is ALKB protein. This is also one found in the higher animals, and it is 1 methyl guanine and 3 methyl cytosine type of damage it repairs. Apart from this, in my next lecture I will be talking about nucleotide excision repair, base excision repair, and mismatch repair. All these contain a huge array of enzymes. Remember, this photolysis itself is an enzyme induced by light. All these repair systems, they are protein mediated, means they are enzyme mediated, and they effectively are involved in the reversal of the damage, or their main aim will be to bring back the correct Watson and Crick pairing, that is AT and GC. Not only that much, AT and GC base pairing has to be restored, but the sensibility of the strand has also to be restored. Means if there is a complete shift in the reading frame, if the reading frame makes no sense, then that also has to be reversed, and that is effectively done by these enzymes. So my next lecture will be on repair of damaged DNA by nucleotide excision repair, base excision repair, and mismatch repair, all in one. Means in the next lecture this unit will be over. So it's a very interesting phenomenon, and it was first initiated by bacteria on this earth during evolution, because they had to sustain the high amount of ultraviolet light and the free radicals which were present on the sea. And this sea, inside the sea, the anaerobic bacteria, that is the reduction reactions, were at large for 1 million 2 million years. I think the reduction reactions were going on, and suddenly they started producing oxygen. So this oxygen amount became so huge, you can say there was an oxygen holocaust. Means there was an oxygen eruption on the face of the earth. And this oxygen, as we see today, also nitrogen is maximum on the atmosphere. Remember this, huge oxygen is very dangerous, nascent oxygen is dangerous. Responsive reactive oxygen species, that is free radicals, are formed in the presence of that. Means anything which is in excess is not good, just like that. And all that was controlled by millions and millions and millions of bacteria, who are still working today, still working in the atmosphere, in the environment, in the C2, and they are bringing about great transformations on the face of this earth. So that is a separate area to be studied, which we study in the nitrogen cycle, carbon cycle, etc. And even nitrogen cycle, you see, it contains a NIF gene, and if there is a damage in the NIF gene, it cannot fix nitrogen. So each and every activity which is pertaining to the DNA, that has a susceptibility of damage, and that can be repaired. That is the point to be understood. So I hope you understood this very interesting topic, and we will continue to end this topic with respect to NER, BR and MMR in my next video. Till that time, as I always wish you to gain more knowledge, become more intelligible, be comprehensive, means understand, remove the ignorance. Darkness of ignorance should be gone away from your minds. There should be more brightness and enlightenment with respect to the genes, genetics and DNA concepts whenever you will encounter these topics in the future. So study well, see it again, take down your notes and become smarter. So that's what I wish. Goodbye. We will meet again in my next lecture. Goodbye.